All right, we're going to learn today how a firefly glows. We're going to tell the story of protein synthesis. So synthesis means to create. We've heard this with photosynthesis. So now it's protein synthesis, to create proteins. So what we're going to do is we're going to start with DNA. That's the first step here, DNA. And then we're going to find out what DNA becomes. So let's come back to that top box. We're going to go investigate how a firefly glows. So we're going to start right here, where conveniently already says start. So we're going to start our story with DNA. Now DNA, we all know, is very important, but DNA has a little bit of a problem. DNA needs to send out all the instructions to build every protein in the body, but it's stuck inside the nucleus. I put that in quotes because being stuck is not necessarily a bad thing for the DNA. It's a very important molecule. You don't want that guy just floating around where it can be damaged or altered. So it's really being protected. But that means there's a little bit of a challenge to overcome. So here's our DNA. Let's zoom into this guy over here. So this is DNA. Right here. And what's happening here is the DNA is being unzipped so that the genes can be read. And a new molecule can be formed that can leave the nucleus. So that overcomes our problem. Before we figure out what that new molecule is, I want to point to this strand right here that's not being read. Funny enough, this is called the coding strand. And the one that gets read is called the non-coding strand. This is important. So we need to know which strand is being read because it will change the outcome of the product if you're reading the wrong one. So how does the DNA overcome this problem? What it does is it sends out instructions via a single stranded molecule. And this single-stranded molecule we call RNA. Similar to DNA, but a couple changes. All right, so then our story is going to take us to something called transcription. So I'm going to change to green. So transcription. We start this process with the DNA. So a molecule called RNA polymerase is responsible for reading that non-coding strand. So that's this right here. It's reading this thing. DNA, if you remember, uses 4 bases, A, T, C, and G. But RNA is a little different. Instead, RNA uses A, U, C, and G. We call this RNA messenger RNA because it's leaving the DNA, abbreviated mRNA. It can leave the nucleus because it is small enough to get out. And it's small enough because it's only single-stranded. So that's important to know. So that's our first process. We went from DNA to RNA. So I'm going to go up here. DNA to RNA. That's our first process, transcription. Okay, now we're going to head down. We can label this as mRNA. So here's our mRNA, and you can see that it can leave right here. It gets out of the nucleus. So this first process happens completely inside the nucleus. We can add a note about that. But now we've left the nucleus, and now the second process begins. So let's choose a new color. Let's go with this nice tropical ocean blue. So now that we've sent out our messages in a secret code, we have to translate it into something the body can actually do something with. So the bases is not enough. So we have our friend, the mRNA. But it gets read by the ribosome. And I'm going to note here that it's read in groups of 3. So think of that like word breaks. It's every 3 letters. So like, so it's read in 3s. All right, so the goal is it makes an amino acid chain. Now, amino acids are the monomers of a protein. So when we have a bunch of amino acids linked together, another name for that is a polypeptide. So I'm going to hop over here real quick just to show you where we're at. So this guy is the ribosome. Now the ribosome is actually 2 molecules. This is part 1. And the guy down here is part 2. And they come together around the mRNA to read it. These here are the 3 bases read as the codon. Okay, but this isn't enough. It's read it, but now what does that mean? How does it actually get the amino acid? And that brings in our friend, the tRNA. So the tRNA has 3 bases on it as well. But these 3 bases are the opposite of the bases on the mRNA. So if they match up because they are complementary bases, and we call that the anticodon, meaning they're opposites. So if they match together, the G with the C and A, that should be T, and then G to C, if they match up together, then this amino acid here will join the amino acid chain. The tRNA will be free to go pick up another one, and the whole process continues. So let's add that down here. So the transfer RNA, it transfers amino acids from itself to the chain. We abbreviate that as tRNA. It has the opposite bases. To the mRNA. And the goal of the tRNA, I'm going to put that over here, maybe. tRNA brings amino acids, I'm going to abbreviate that AA, to the ribosome. So mRNA has a lifespan, but it can be read more than once before it expires. So I'm going to hop back up here. So I went from DNA to RNA to protein. This is central to life. So we went from DNA, used transcription to make RNA, and now we've used translation to make protein. But we're not quite done yet because we just have this protein. Does that actually mean anything? It doesn't unless we follow it all the way to the end of our story. So we have all these bases. We've read them. Now we have all these amino acids in the chain. When we get up here, what actually happens is they make a protein. And that protein is something called luciferase. It has an ASE ending, which means it's an enzyme that speeds up or catalyzes a reaction. And luciferase makes light. So it's not just enough to make the sequence of amino acids, they also have to be folded correctly, and then they can make this process. So we have successfully made our light, so we can go ahead and color in our firefly and make him glow. Last but not least, I'm going to change colors. I'm going to jump down here in the dark blue. Maybe. We're going to come down here and we're going to practice transcribing and translating from DNA to mRNA to the protein. So we have ATG. So we know that A would normally go to T in DNA, but instead it goes to U. T goes to A, and G goes to C. C goes to G, G goes to C, and A goes to U. You're going to continue that whole process. So I'm going to jump ahead right now, and I'm going to open up this codon wheel. I prefer the wheel. You can use the wheel or the chart, whatever makes you happy. But when we look at this, how we're going to read this, I'm going to take a screenshot so I can write on it, is we start from the very center. I'm going to use black so it's easier to see. So our first base is U. That's right here. Our second one is A, so we move to the next out of the circle, A. And then we move to the next one, which is C, right here. So that tells me our first amino acid is TYR. That's its 3-letter abbreviation. So we came dangerously close, though. If the base had been an A or a G instead, UAA or UAG, we instead would have had something called a stop. And if stop shows up, that means the amino acid doesn't get read any further. It just stops. Nothing else will happen. So those can be quite tricky. So let's jump back in over here. So that tells me that our protein becomes TYR. It's not going to let me write on it anymore. Okay, so that becomes TYR. Okay, I guess we can't write on that anymore. All right, and that is how a firefly glows.